Welcome to the AI Research Roundup. I'm Alex. Today we're looking at a paper from the Hugging Face trending list published on September 23, 2025, just yesterday. The striking result is a simple, flow-matching transformer that folds proteins competitively without multiple sequence alignments or triangle updates, reaching TM score 0.837 mean and 0.916 median on Cameo 22. The paper is titled, SimpleFold, Folding Proteins is Simpler Than You Think. And as we'll see later, the implications for model design are quite significant. Beyond accuracy, it generates realistic structural ensembles, scales from 100 million to 3 billion parameters, and runs efficiently on consumer hardware. Alright, the previous segment highlighted the core idea. Figure 1 shows what that looks like in practice. Panels A and B present predicted structures that align closely with ground truth, with high TM and GDT scores. Panel C illustrates an ensemble for 6 NDW, which captures flexibility across plausible conformations. Panel D plots CASP14 GDTTS against forward gigaflops, indicating better accuracy as model size grows. Panel E reports inference on an M2 Max 64GB laptop, where time scales with sequence length, yet stays practical. So Figure 1 showed outcomes. Figure 2 shows the streamlined architecture. A pre-trained protein language model provides sequence embeddings, which feed an atom encoder, a residue trunk with grouping and ungrouping, and an atom decoder. The same transformer block repeats across stages, with time-conditioned scale and shift layers, so the network learns a flow that maps noise to structure. A skip connection preserves atom identity, and local attention keeps computation efficient. Crucially, the design avoids pair tensors and triangle updates. Okay, Figure 2 detailed the architecture. Table 1 presents head-to-head accuracy on Cameo 22 and CASP14. On Cameo 22, SimpleFold 3B reaches TM score 0.83, 7 mean and 0.916 median, matching ESM-Fold and close to AlphaFold 2. On CASP14, it posts 0.720 and 0.792. The table also shows steady gains with scale and clear wins over ESM flow. So, the previous table covered accuracy. Table 3 reports two-state conformation results for ApoHolo and fold switch ensembles. The metrics track residue flexibility and ensemble TM score, which reflect how well predicted ensembles match experimental states. Sequence-based baselines lag behind, while MSA-based models are strong. In the hours block, scale helps markedly, and SimpleFold 3B leads with ensemble TM scores of 0.893 and 0.916 for ApoHolo, and 0.734 and 0.766 for fold switch. So, the previous table covered two-state ensembles and showed clear gains with scale. Figure 4 presents scaling behavior across 6 panels. The left panels relate training gigaflops to GDTTS and TM score, and larger models steadily score higher because added capacity and compute raise accuracy. The middle panels chart training steps, which lift both metrics with longer budgets. The right panels vary data size, and the largest 9.2 million set delivers the best curves. Alright, the previous figure traced scaling. Table 7 reports inference time on a single H100 across sequence lengths. The comparison shows SimpleFold runs fast, especially for long sequences, because the transformer is lightweight at inference. At length 1024, AlphaFold 2 takes about 111 seconds, AlphaFlow about 372, and ESM-Fold about 44, while SimpleFold 100 million finishes in roughly 5.6 seconds at 200 steps or 14 at 500. ESM2 adds negligible overhead, time rises with steps and length, yet remains practical across SimpleFold sizes. Wrapping up, 2 takeaways stand out. A plain transformer with flow matching achieves competitive folding accuracy without multiple sequence alignments or triangle updates, and the generative setup yields realistic structural ensembles while running efficiently on modest hardware. The broader implication is a simpler, scalable path for protein modeling and deployment. That is it for this episode of the AI Research Roundup. I am Alex. Thanks for listening.